Okay, so hi everyone, and welcome to this third webinar in the OBON series. We'll have two presentations by OBON endorsed projects, but I'm going to start with a short overview of OBON. Apologies to those who may have heard this before a few times. It's been kind of the standard presentation in these webinars, but we thought it would be useful to set the scene for anyone who's new. So I'm Sophie Siave. I'm the CEO of POGO, the Partnership for Observation of the Global Ocean, which is the lead organization for OBON. And we also have with us Margaret Lynan, who's the chair of the OBON Scientific Advisory Committee. So I'm just going to navigating to two screens here. Okay, so this is a link to the OBON strategy, which was released earlier this year. And so you can download this and have a closer look. But in a nutshell, OBON aims to establish a global biomolecular observatory for a sustainable ocean. And our vision is to accelerate informed decision-making to restore the health of our ocean using the universal signatures of life on Earth, biomolecules. And our mission is to create a hub for biomolecular measurement of marine life. We have four main objectives. These are quite detailed, but basically number one is to build a coastal to open ocean multi-omics biodiversity observing system through the development of autonomous sampling and also promoting methods and technique development. Second objective is to enhance marine omics data systems and enhance ecosystem models. Third is to partner with marine conservation and management specialists, policymakers, the private sector, and in doing so to address sustainable development. And the fourth, although these aren't really in kind of consecutive order, is to develop and transfer capacity through training programs combined with funded equipment programs supported by development and aid agencies and philanthropy. So these objectives can be summarized in this diagram, which sort of centers around the development of networks and resources, the innovation and technology development, and enhancement of information use through models and digital twins, and with the ultimate goal of informing a sustainable future. So OBON is a UN Ocean Decade program, and as such, we endorse projects kind of underneath the umbrella of the program. So we've participated in quite a few of the decade calls for actions so far, and we now have 20 endorsed projects underneath OBON. And this webinar series is really designed to introduce and provide updates on all of our projects. So this is a map of the sampling locations where the OBON projects are making biomolecular observations. Most of these are fixed time series stations, but there are also some one-off sampling sites in there as well. And so we're gradually expanding our coverage, although we still are very much focused on coastal observatories at the moment. But we keep participating in the calls for new projects and encouraging more open ocean initiatives to join us. So if you're involved in or aware of any initiatives that could fill some of our gaps, for example, in the Pacific, in the Southern Ocean, around Antarctica, please get in touch with us. So OBON is organized mainly through Coordination Office, which is currently hosted by the POGO Secretariat, and Luz Rodriguez, who's here with me, is the program manager for OBON, and the POGO Secretariat also supports the program in various ways. We have a scientific advisory committee and an executive committee that meet regularly to keep the program advancing. And as I mentioned earlier, Margaret Leinen is chairing both of those committees. We're in the process of setting up a user advisory committee to help us with user engagement and sort of the policy connections that are very important for the program. And we have several working groups that are currently at different stages of development. Some are still being planned. We've held a number of workshops. We had a very successful workshop at the IOC Westpac conference in Bangkok earlier this year, which has really helped us to expand our network towards Asia and the Pacific. We're planning a workshop on eDNA for fisheries management, being planned for next April or May. And we're also planning to have a workshop on capacity development and setting up an eDNA network in Latin America in Colombia next year, hopefully. So that's a very brief overview of OBON and some contact details there. If you wish to get in touch, and particularly if you're interested in engaging and working more closely with OBON, we would very much welcome that. So I will now hand over to the first of our speakers, which is Jennifer Sunday from Canada, who will be presenting. I'll let her introduce her project on her eDNA observatory work over in Canada. So Jennifer, the floor is yours if you'd like to.
Excellent. Going back to the screen sharing that we practiced. No, one more try. Great. So
perfect.
Great. Hi everyone, I'm very happy to be speaking today, and thank you so much to the OBON organizing group for arranging this. So I don't have too much. I'm just going to give a big update on the PECO, the Pacific eDNA Coastal Observatory. We joined OBON, I think, just near the end of 2022, but we've been running our program from 2021 until now. So it's an annual program, as you're going to see. Here we go. So the focal area is on the Pacific coast of North America. And our project is a networked collection of seawater for eDNA that happens at a snapshot. So in between about 2 months, every summer, the network of partners go out and collect the seawater. We send kits to them. They send kits back to the Hakai Institute, which is located just up in here on the east coast of Vancouver Island. And all of the samples are processed for right now. DNA is extracted and is processed for fish markers, so 12S. So this is just a big summary. We have 16 regional nodes. There's actually about 18 different partners because some nodes are broken up by partners. We have over 70 sites. We're the first that I know of a coordinated eDNA observatory in North America. And every year we have over 50 participants. Our partners bring out their trainees, and everybody, lots of people participate. So that's the big overview. And if you look at one of these regions, what we do is we ask every partner and we help co-design how they choose sites. And so they go to 5 different sites ideally, and collect 4 samples in each site plus a negative control. And so that the goal is to cover the fish regional species pool. We're focused on one habitat type, which is seagrass. So it's seagrass or eelgrass. So it's currently limited in just that habitat scope, but some of our work is finding that that's going to cover a lot of the fish in these little bays and inlets for the region. So we're really trying to capture the regional fish biota so that we can track their geographic ranges across this region and then over time through every year. And so here's we summarize just some of our successes. So I see this as a success. We have deployed this network for 4 years now. In the first year, we collected both eDNA and a fish capture technique, which is beach seine surveying. So in these seagrass habitats, this is a fairly good way to collect the local fish just right inside that site at that moment. And so we can ground truth what we're finding. And then we've deployed the eDNA, not every site every year, but we're still covering that by a geographic range. And so it involves a lot of partners. The partners are, for the most part, university researchers who have been working or monitoring in these eelgrass beds for a long time. But we have a couple of community groups, and we have one of our partners has been the Haida Nation. So we've been really happy to be working with those partners up in Haida Gwaii. Okay, I'm going over this at the very high level, but another, so here's sort of just the view of how we do it. We collect from the field, the samples are processed in the lab. Those results are further processed through bioinformatics. And then we see this very important step, which is the biodiversity analysis that we need to improve knowledge. And we consult with sort of knowledge users and what sort of information they need so that we can make sure we're analyzing to that level or learn about maybe what we should be doing differently. And so we're happy to have been working with OBON and to try to get all of our protocols on protocols.io. So the Hakai Institute is already way ahead on this, and they had already published the protocols for the DNA extraction and the metabarcoding prep, and we've added our protocols for the field sampling. These little construction cones represent that this part is still ongoing. So I'll show you some data from some species and some through the ASVs and the species and their biogeographic distributions. But our goal is to have these all available and also interoperable. So getting these all into the big global databases. And then also to do the biodiversity analysis. So this is just a schematic, but the people involved for the sampling, of course, it's all those 50 people. And Sue Velasquez from the Hakai Institute runs all of the kits, deploys them, gets them back, bleaches everything. So it's a big operation. Matt LeMay and Ruth Cavallo do oversee all of the genomics at the Hakai Institute. And then these orange cones here, this is Kate Sheridan, who's also on the call, who is working really hard to have a reproducible workflows that we can share for all of the bioinformatics and then the analyses. Okay, so here I promised a little bit of data. So this is from 2021 and then 2022. On the x-axis are the fish species, identified 2 species. So each one of these little ticks is a fish species. And on the y-axis is their occurrence at a site that is just spread out across latitude. by rank. So the idea is you could see, if you look at the little map here, you can see that you can see some patterns of biogeography where you have species up in the north that are different than the ones down in the south. If you look at any fish species, we're working to see if these species distributions are meaningful, if they're really good species distributions, and how many samples do we need to see those distributions, and maybe for which fish is it working better. And then if you look across, then you see a community. So we can look into what is happening inside that community, what the network looks like, what the interactions of those species might be, and then eventually how they're responding to environmental pressures through the years. Okay, some other successes. We had some really great consultations this year with knowledge users, and one of the big ones was this webinar and workshop that was organized by the Ocean Decade Collaborative Center for the Northeast Pacific. And what we learned from these consultations was that a lot of the community and Indigenous-led monitoring programs in this region, they really want to track species of interest with high confidence. And so it was a bit of a pushback on the metabarcoding approach because it made us sort of think, okay, what else do we need to do here? And we're going to probably add qPCR with species of interest through the samples we've already collected so that those can be easy pickups and use and this will hopefully help with engagement. So we can add some more groups as well, because this is what they're really interested in. Another process, this is through another project that's in Canada, where we've been working with Fisheries and Oceans Canada, and we heard from them last week a little bit from some of the partners of this project. And one of the questions that they really addressed was, how many samples do we need if we're going to detect biodiversity change? And I think we talked about this a little last week. And so one of the things we can do in any system is we can characterize the shape of the accumulation of how many species we see in a system given how many samples we take. And we expect diminishing returns in a curve like this. And we can do this for our system, and we've been doing that. So we can sort of identify how many samples, what's the likelihood that we're missing species, and given this technology, and how many samples do we need? But the other thing that we can do is ask this second question, which is, how many samples do we need to detect differences over time or over space? And for this is a project that Eden Tekwa is leading, where we're trying to ask, trying to sort of characterize this relationship and say, look, as we have more samples, we have more confidence in differences that we're seeing. And then we could characterize this curve, which takes a little bit to think about, but it's as you have more samples, you can detect more subtle changes. So the detectable differences on the y-axis here, you have more power, or you can detect smaller changes. If you have fewer samples, you really can only, with confidence, detect big changes. So 10% of biodiversity of species richness, for example, instead of maybe 2%. Those are the changes that you're in a position to be detecting confidently. So we're hoping this work is useful broadly. That's why I wanted to share it. And there'll be a package, and there should be some reproducible parts of that. Okay, so we were asked also to think about how how we might benefit from the group and where we need help. And I think for our project right now, and we've talked about this a lot, but I think our project is now ready to dig in, is we want all of our data to follow FAIR principles, but now is the time to actually put the rubber to the road and figure out where are we going to put our data. What are the data standards? We're ready now to be making sure that we're matching our data standards. And then this is one I really wanted to talk about, which is some of our partners are interested in a system that can give permissions per use request down the road. After we've sort of maybe published the paper, we want the data to be up, we want everything to be out there. they still are interested in a toggle where they could say, some of the partners, well, I don't know, I want it to be for this use and not for that use. Can they give a permission? And I think for some of the community groups and the Indigenous partners, this would be an important piece to talk about. So that's what I wanted to bring to the group. And that's all. There are many, many, many people to thank. Many names are listed here. I've just highlighted Margot Hessing-Lewis and Matt LeMay, who helped to run the program. And I talked about Kate and Rue and Sue, who are doing a lot of the legwork. Thank you.
Okay, great. Thanks very much, Jennifer. I don't think there are any questions yet, but feel free to put your hands up if you do have a question.
Sorry, sorry, Nick and you. There are two hands.
Okay. Yes, Nick.
Thanks a lot, and thanks, Jennifer, for a really interesting talk. I actually have several questions, so I can just do one at a time and let others people have. But my first question and my top priority is, you mentioned that for your first year of study, so 2021, you were doing both purse seining off the shore and your eDNA. Could you say anything about the results you found and how it correlated with one another?
Yeah, I could actually. Why don't is it okay if I show another slide? Yeah, they're good. I had a slide. Maybe I won't try to do that, but so it's beach seining, not that just to create. It's just like we sort of drag this the net along the bottom, so it goes top to bottom, catch fish. I really want to show you the graph because so we make these little Venn diagrams for every site, right? And so we can see there's lots of overlap. It's probably about 75%. So it's really on par with what people are seeing in other studies of the fish caught in the beach seine to what's in the eDNA. 75%. Okay, so are we happy about that? And so we're like, hey, what are we not seeing in the beach seine that's in the eDNA? What are we seeing in the eDNA that's not in the beach seine? And I've had a couple of students in our research group who have been really diving into that and saying, okay, we can list the species, but what are their traits? What can we make any sense of this? And so it's often, and we have this in our PECO data as well, the species that are not getting picked up in the eDNA are very clearly, most of them are low biomass when they're in the beach seine. So they're just a low biomass, I'm going to tell you, few number or small body size. The fish that are being picked up in the eDNA and not the beach seine, as you can probably expect, I could even ask you, what do you think they are? But they're the fish that are either they come through in big numbers, but they just don't happen to be there when you draw the net, or they're local, they're abundant just off in slightly different habitats. So they're just not in seagrass habitats as adults. Or juvenile. They're not being caught in the beach seine, but we know their habitats they prefer to be either deeper or a little bit offshore. So, and one of the projects we've worked on is, as you look across space and you look across habitat types, how can you explain what the difference between what you're seeing in beach seines and in eDNA? And the way I've kind of told the story summarizes that work. So we have a little paper coming out soon, which I could share with the group that's about how how the grain size of a beach seine survey is small, but it means you see lots of differences in the fish and they're very closely related to the habitat type. Whereas the grain size in the eDNA is just that much bigger that you don't get to articulate so much what you see between every habitat, but you see the more of the regional fauna. So we love that because we want to look at regional biodiversity, regional species pools, but we know we're not going to have a great power to detect differences between sites or habitat type. Long answer.
Thank you. We'll come back to you in a bit, Nick, but we also have a question, comment from Senji Lin.
Yes, hi, Jennifer. That's interesting work. My question is about the quantitative. I just wonder what is the real purpose, or what can that qPCR do? And I guess my point is, for eDNA, I expect to see a lot of variations depending on like what this organism does, not just the the number of individuals or the abundance, right? Whether they only shed a little piece of the skin, or they actually like pee there, or do whatever. So how can you use this quantitative data?
Do you mean abundance? Is that what you mean?
Yeah.
How could we use it to give a signal of?
Yeah, I thought that this eDNA is, I would be more confident in using it as like absence and presence, but in terms of quantitative, I just wonder. Yeah.
Yeah, I mean, that's a great question. And there's lots of people in OBON that are thinking about that as well that could probably answer. But so the way I've presented today, it's all presence and absence. So we can very easily use the presence absence data. I come from a biogeography background and species range shifts, and we use occurrence data. So it's just presence, non-detection, and you can say a lot just from that. But how I've seen people using either the read number that comes from when you do metabarcoding, you get an output of read number. We're trying to be careful not to take the relative read numbers literally. So if one species has a high read number and the other has a low, that could be a reflection, like you say, of all sorts of processes that don't reflect abundance. But if those read numbers change over time for the same species, or if for the same species that the pattern of the read number changes across space, we are kind of interested in using that, because we think we can use, and it's been shown a little bit, that the relative read number within a taxon, you can kind of assume that a lot of the same processes are leading to its read number being low or high. So, but we haven't started using it that way. But I think that's where there's possibility in doing that. And then some labs are working on. I know Ryan Kelly's group in Washington, University of Washington, are working on getting in there and saying, look, I know how metabarcoding works. I know they're competing for space in that PCR reaction, but they are actually working to try to still use the read number from a metabarcoding result to detect relative abundance, but that's still ongoing.
Yeah, it's just because if fish, they swim, they come and go. I could kind of think of if it's like a sexile organism, it's like plants or benthos, they are always there. Then maybe this quantitative data could reflect about the fluctuations of abundance, because they cannot really go away, things like that. Yeah.
Yeah.
Well, anyway,
we
just
think
a thought
about the
maybe.
swimming and sessile difference quite a bit also. That's a really good point. Yeah.
Okay, great. Well, thank you.
Thank you. Nastassia has also added something in the chat in relation to this discussion. Are there any more questions before we move to Nastassia's presentation? Nick, did you want to come back with another question?
Only if there's time. I can also wait till after. I don't want to take away too much time from the next presenter, so I leave it up to you whether there's time.
I think we can take one more, because in theory we have an hour and a half. We may not need to use all of that. And I don't want to delay too much, but go ahead if you have another question.
Yeah, so it was regarding your work to see how many samples you needed to detect major changes. I just wanted to make sure I understood correctly that your survey was once per year, right? You were sampling 4 different sites did I understand that correctly? You only sample once a year.
Curves,
so it's like,
that's absolutely right. Yeah. So the curves, those cartoon curves that I was showing you, we're populating those with other data sets where we've got many more samples. And in fact, that was this work of this big genome project where we tried to oversample. So we said, let's take 600 samples and see what happens when we subset them. 200 sites, 3 samples per site, and then we're trying to just learn how do we, assuming we saw everything by that point, which we already know we didn't, but so we can characterize. It's really interesting. The whole project has gotten us into realizing when you survey biodiversity with any tool, you've got your technology, it's got a detectability. You also have the heterogeneity of the region you're trying to characterize, spatial heterogeneity. And if you have a more heterogeneous habitat, you're going to need more samples. If you've got a more homogeneous, sorry, if the biodiversity is more homogeneous, you're going to need fewer. We get that, but it got us into this concept, and Eden is leading this, of describing what are the effective number of sites in a region. And if we know what so is 200 the effective number of sites? Is it 600? How many, with our technology, how many sites would we need to characterize all the things that technology can detect? And so that's helped us to think about, like, we sampled 200 sites in this big project to characterize a regional fauna. We think, based on the biodiversity modeling, that we probably needed 600 of the same types of samples to see everything, everything. No false negatives. But then that helps us to say, well, now we know if we only had 5 samples, we know where we are. If we took 10 samples, we know where we are. And it's not too bad. Like, it turns out that it's looking like, at least for our region, which is Northeast Pacific, if you took 20 samples, you're probably able to detect 10% of species richness change. 20 to 40 samples, somewhere in that ballpark. 5, 5, not so good. So when we say we take 5 samples, we're going to 5 sites, and we're taking 4 samples in each. So we think we're capturing 20, but they are spatially grouped. So it's not something in between.
Thank you very much. That's really interesting. I mean, we always have to make sacrifices like that. I mean, everything has to be scalable. So whereas it'd be lovely to do 600 samples everywhere, very few of us are actually able to put in that amount of effort, except if we're just baselining. But for that purpose, I think this is really useful. So thank you very much, Jennifer. Thanks
for your questions, Nick. And I see Nick Jeffries in the audience, and Nick Jeffries on that project I was just describing as well. Hi, Nick.
Okay, and there's also a question from Erin. Did you notice that these sample sizes, sorry, I'm struggling to read, I've got the screen miles away, change depending on the variability of the system in a given year?
Well, yeah,
for example, do you need more samples in more variable year?
So let me just think about that for a second. So if I mean, we're trying to describe how you can make, if you sample a system and you say, here's how many species there are, here's what the species are, you are not missing species, or you know kind of where you are between what you've seen and what you've missed from one given sample. And so if you want to detect change over time, then I guess we're arguing that you would have to try to get precision at both time points so that you could detect that change over time, if you're interested in saying something about the effect size of time or what happened over time. But this is about temporal variability. So just like maybe from week to week or from day to day or hour to hour, if the system is really variable, that's kind of more variable years. Do you want to clarify, Erin? Do you mean like?
Yeah, no, I'm sorry. I was just thinking, as you were describing it, made me think, and thanks for this, about the graph or the graphic you showed of the composition across latitudes. And I was just thinking that, wondering if in years where you might have extreme influence of equatorial water or, waters coming up, like the composition would change dramatically. And so I was just wondering if how your sample size might change it depending on sort of the conditions of a given year, or
Right. Yeah. Well, okay. So that helps. Thanks for clarifying. So to me, that these years are then sort of like anomalous, or there's some change happening within those years. I mean, it could be that you needed fewer samples that year. If it's like something takes over and there's fewer, the diversity is lower and there's more homogeneous, then you could see, it could be that actually you might have gotten away with fewer samples. And then you'd still be able to detect change over time. I mean, one always wants more samples. You can always say you should sample more. But I don't think that really, I mean, I think this is the point, is we're trying to say, how could we detect biodiversity change across those 2 time points? Even if we didn't know the system was going to change, we just sampled and we wanted to see change over time. But anyways, absolutely. When you look at the pattern of fish diversity that I was showing, we essentially only have 1 sample, which is that year. Now we're going to have 10 years, we hope, and so we're going to see this pattern of change over those years, and try to associate it with drivers. The drivers will be, I mean, they're local. We'll have all sorts of environmental pressures happening locally, but then also some, like you're describing, large scale coastal system changes that effectively we only have 1 sample of. So I hope that's why we're replicating everywhere, right? We've got the network of networks, so we can start to make sense of the patterns we see more broadly. Our way around that problem is to come with hypotheses. So we're going to say, like, what do we think is going to happen? What given where this fish is located and everything we know about that fish. So Kate's been working on a big traits database, and what temperatures is that fish normally found in? What do we think should happen from year to year? And then did we see that? So at least we've got a little bit more grit in our analysis. I wandered away from your question. But thanks for asking.
Great, thanks. Thanks very much. So we'll move to the next presentation. If there are any further questions for Jennifer, please put them in the chat, or we might have time at the end to come back when we have our sort of general discussion. So thank you very much. And Nastassia, if you would like to share your screen, we're going to move south, staying on the west coast of North America, but moving a bit further to warmer climes. And Nastassia will present the West Coast OBON project, which was endorsed quite recently by OBON. Okay, Nastassia, the floor is yours.
Great, thank you.
Just put into presentation mode.
Oh,
that would be great.
Great. Is that, can you see that okay?
Yeah, perfect. Thank you.
Wonderful. Well, thank you so much, Sophie and Luz and the OBON organizers. It's really great to be able to present to everyone today. And thank you, Jen, for a really fascinating talk on PECO. I really would love to talk more with you about it. As Sophie mentioned, we're actually staying in the same geographical region as the West Coast Ocean Biomolecular Observing Network, but a very different type of project, I would say, a very different structure and organization. And so hopefully I'll be able to share some different types of OBON programmatic goals with you. And we did start as a Southern California-based network. We were the SoCal OBON, and then as it evolved, it became clear we had some natural alliances and linkages among programs that made it just silly to limit ourselves to Southern California, and so it became West Coast OBON. And our overarching goal, as stated here, is integrating and harmonizing molecular observations in support of sustainable marine management. My name is Nastassia Patin, and I work for CalCOFFE, the California Cooperative Oceanic Fisheries Investigations, and the Southern California Coastal Water Research Project. So I have a joint appointment at these two with these two programs, both of which have their own observatories, if you will, their own monitoring programs. And so this is a general map of our geographic scope, and as you can see, this is very similar to the scope that Jen presented in her project. But we are, rather than being one observing program, or one sort of monitoring program, I should say, where we have folks going out sampling and we combine all the samples and present data We are actually more of a network of networks, a collection of institutes and existing monitoring programs that we are bringing together to begin to harmonize, so that we can harmonize our methods and adopt some of these standardized approaches to all of the molecular observations so that we can begin to integrate the data sets and really leverage work from independent groups so that we're not all sort of working in these siloed isolations. And so this is hopefully a slide that just shows that big picture idea where we hope to connect data, where people are monitoring biodiversity at all different trophic levels. And we hope to start to bring those data sets together and create these holistic big picture ecosystem dynamic observations. But to do that, we really need to harmonize our methods and facilitate this adoption of sort of best practices and standardized approaches so that we can compare apples to apples data sets. And so along this West Coast, who's included in the West Coast OBON? We have Cal Coffee, which I mentioned in the intro slide, and we are the longest marine ecosystem monitoring program in the world. We go out quarterly to survey all the way up from San Diego to Northern California, including offshore areas. And we've been going for 75 years now. We have our 75th anniversary this year, which is very exciting. There's also the Southern California Coastal Water Research Project, which runs the BITE monitoring program, which is the Southern California BITE, which is this area south of Point Conception. We have the Southern California Coastal Ocean Observing System, or SCOOS, which is part of NOAA, but involves various different observatories, including peer-based observatories along in Southern California. The California Current Ecosystem Long-Term Ecological Research Program, or CCLTER, which is more of an academic program funded by NSF, and also doing ecosystem monitoring on something like every 2 or 3 years, I want to say their cruises are. We also work with the NOAA's Pacific Marine Environmental Laboratory, which runs the West Coast Ocean Acidification Cruises, which are West Coast-wide cruises that have traditionally focused on ocean acidification parameters, but are now including eDNA as well in their sampling program. And MBARI at the Monterey Bay Aquarium Research Institute and the Hakai Institute are two of our more independent kind of nonprofit partners that also run their own sets of observations and monitoring. And so we at the West Coast OBON, we are this network of networks, this conglomeration of groups that are going out with the common goal of observing biodiversity in the ocean, and we are bringing everyone into the same room so that we can start to intercalibrate and intercompare our data sets. And so part of the motivation for this is that there is just increasing evidence of these sort of system-wide effects that we're seeing, where what happens in Southern California is going to be affecting what happens off the Pacific Northwest. This interconnectivity of the California current and just the oceanography of the Northeast Pacific Ocean makes it a very natural or important effort for groups that are measuring in small areas along the coast to come together so that we can start to integrate these observations and understand things like marine heat waves and how the effects that are being felt in British Columbia might, translate to effects that we see in Southern California, whether that's red tides or hypoxia or things like this. And so that was a big motivation for expanding the geographic scope from SoCal OBON to West Coast OBON. Okay, so I've mentioned a lot about aligning our methods. So this is one of the big objectives of the West Coast OBON, and this includes everything from sampling collection, frequency of sampling, experimental design, all the way through to bioinformatic analysis and metadata standards. And so the hope is that by aligning these methods, we will be generating data sets that, as I said, can be combined, compared, and used together to form this holistic overview of the ecosystem. So one of the first things that we did with the West Coast OBON was to assemble this list of recommended protocols. And so this was in concert with several of the different our members who are working on improving, developing molecular methods. We sort of aggregated the most, I would say, popular, but also most validated sets of primers and PCR protocols for various marker genes, so that folks that are maybe new to the field or wanting a starting point for how to move forward with their metabarcoding assays could use this as a resource. And so we have this, it just exists as a Google Doc, but anyone can access it from our website. And we are always revisiting it and communicating with folks that are in the trenches on these development efforts. But we hope that this is a good starting point for many people that are hoping to go out and do some eDNA-based observations. Another big push that we have been working on is generating a library gap analysis for our region. And so this effort was really led by Zach Gold at PMEL, but he contacted, gosh, I don't even know how many monitoring programs on the West Coast. This says 25 plus. I think it was probably close to 50 by the time he was done. But there are just so many small scale monitoring programs run by NOAA, universities, nonprofits that are interested in understanding marine biodiversity. And so he contacted all of them and generated a list of most valuable monitored species. And then using an automated computational or automated programmatic pipeline, he could systematically check both GenBank and BOLD for DNA reference sequences, linked to this list of priority taxa, and then using that information could do this gap analysis, letting us know what is and is not present already in the databases and what we should focus on maybe getting in there. And so what we found is that about 50% of species that are considered high priority monitoring targets have no marker genes in GenBank. And so this was a really valuable finding and is guiding us forward as we think about how we start to improve our reference libraries and prioritize efforts on that front. And so sort of continuing on in that vein, we came together for a workshop in May of 2024 with the specific goal of generating a best practice document for generating DNA reference barcodes. So everything from sample collection, specimen collection, vouchering, archival, to DNA extraction, sequencing, and data curation and analysis. And so we had speakers from large collections programs, including the Scripps, benthic, and pelagic invertebrate collections, as well as the Los Angeles Natural History Museum, to talk about how they go about collecting and vouchering specimen organisms that can then be used as the basis for your genomic DNA, where the reference barcodes are generated. So that was really valuable. Just speaking for myself as a former marine microbiologist turned sort of eDNA scientist, I'm never been on the side of collecting worms and putting them in ethanol. So it was very interesting to learn about that process and how what are the guidelines that need to be followed in order to ensure that the sequences you're generating are high quality, are accurate, and can be linked back to your voucher organism. So we had some talks on that topic, and then we also had bioinformaticians from the NOAA Omics program speak about different approaches to sequencing, and with a real focus on sequencing entire mitogenomes. So rather than just amplifying a single marker gene, which may or may not be used in 5 or 10 years, if we come up with something better, we're really emphasizing the importance of sequencing entire mitochondrial genomes, which can give about 20 times the data for 4 times the cost, and will exist as a sort of repository for gene mining and gene analysis, regardless of what marker you're looking at, as long as it's in the mitochondrial genome, of course. So, so that was really, I think, really valuable, really helpful workshop. And what came out of that was a document that is close to being finalized. We're still getting some feedback from some of our subject matter experts, I would say, but we have a really robust guide that takes a user from specimen collection and all the metadata and effort that has to go into the collection all the way through our bioinformatics and data reporting process. And so we are hoping to get that published this winter, put it live, and make that another resource for the community so that we can sort of facilitate and accelerate this reference library expansion, which I think, as we all know, is a real hindrance to good biomonitoring in the ocean. We also work very closely with BBOP, which I think you may have heard from last Sunday in a webinar. And so BBOP is the best biomolecular ocean practice. Anyway, they better biomolecular ocean. Someone help me out here. They have the templates for protocols with some minimum information required that would be considered like a rigorous, high quality protocol. And they have provided these really excellent protocol templates so that we can start providing our methods for, again, anything from sample collection to water filtration to PCR amplification of marker genes, sequencing. These can all be standardized protocols that exist on our West Coast OBON repository and provide a guide for all our members to start to harmonize our practices. And so that's been a really fruitful collaboration. We're really grateful to BBOP and hope to continue working with them. So the directives for this this presentation were to talk about challenges and collaboration opportunities. And so I think it would be remiss of me to talk about challenges without noting a lack of funding for these projects, which just means for us, we don't really have support for the coordination of in-person meetings. And so we've managed to piggyback our workshops and meetings onto other large meetings that we have. So Cal Coffee hosts our annual conference, and we've now twice been able to add on an extra day and have our West Coast OBON members join because of the existence of that meeting, which has been really fruitful. But it is, I would say, a challenge to bring together this large network of organizations that doesn't sort of naturally convene regularly, so that we can all move together with a united front and have the really valuable types of meetings and discussions that we try to have that move our science forward. And so while we look around all the time for informal or external, I should say, sources of scientific collaboration, sources of funding, it is definitely a challenge that we face to keep our group sort of more cohesive. And then both a challenge and a strength, I would say, is the diversity in our community members. And I mean that in a number of different ways. And so we have scientists that span a real range of expertise. So everything from molecular biology to fisheries science to marine ecology, cellular and organismal biology. These are all members of West Coast OBON, and they really provide an incredible foundation for, I think, a holistic science, and make sure that we're all sort of working in accordance with field-specific best practices. But they also span a range of professional fields. So we have academics, federal scientists, nonprofit scientists. And I call this a challenge because as hopefully everyone on this call is aware, experts in their field can have very strong opinions about how things should be done, and what is considered optional versus obligatory. And so I think developing this guide on reference barcoding has really showcased some maybe where there are disagreements among members of the field, or opportunities to really develop these best practices. And so it just means that reaching consensus can be slow, but ultimately I do think it is a strength of our group and makes our products more rigorous and more informative. So I wanted to highlight that. Finally, are some collaboration opportunities. So on the right, this is a flyer for the Ocean Observing Conference, where I mentioned we piggybacked our May 2024 meeting on the back of this conference. And this was really great because it already was a natural opportunity for scientists from all of our networks to come together and be here in person. And so we are constantly trying to look for opportunities where we can work together in a more formal way, collaborate, share ideas. And so we're trying to get creative with finding avenues for doing that. I think there are certainly some opportunities to identify and share potential funding sources and grants. This is, again, I think the scale and the diversity of our network is just it's just a challenge to identify all the areas of potential cross collaboration. But definitely, we've already had some sort of informal linkages that have been really nice, and we hope to continue those in the future. And finally, we would like to improve our communication and outreach to help other OBON projects as well as ourselves tell our stories more effectively. And so we're looking at a diversity of media types. We work a lot with, for example, the Scripps social media or media office, I should say, just because that's where Cal Coffee is hosted, and it's a easy avenue for doing that. But we would love to expand the range of that and maybe leverage our OBON partners to improve some of those communication and outreach efforts. So I just mostly want to acknowledge the 3 co-leaders of the West Coast OBON, Zach Gold from NOAA PMEL, Susie Thoreau from Southern California Coastal Water Research Project, and Aaron Satterthwaite from both Cal Coffee and Sea Grant. And really, it's just a joy to work with all of them. And I think it's the diversity of their views and backgrounds has been a real boon to the West Coast OBON. And here's a little QR code that will take you to the website. I personally am not on Twitter, but I am on Blue Sky. There's my handle, and we're really happy to talk to anyone who's interested in our network and our program. And I'll take questions.
Great, thanks very much, Nastassia. I think in OBON we can identify with most of the challenges that you've presented of lack of funding and having to, find funding for meetings and needing to piggyback onto other things. So I think that there's a lot that, OBON can learn and maybe help other regions with kind of based on your experience and the way that you're starting to do things, and hopefully OBON can also help you. There was a question, it's a bit further up in the chat from Becca Millard. Are the scripts for species gap analysis available for others to use on their species lists in other regions?
Yes, thank you for that question. So the list itself of priority species is somewhat region specific, but we're happy to share that if it's useful for another region. And then the code for doing the gap analysis, I believe that is currently not, it's not public like on GitHub or anything like that, but it is a goal for us to make that available. It may actually exist in some form, like it's in a sort of under development, but I think our final workflow is not yet publicly available. But we would like to make that, yes, available to anyone. And so I will make a note to check the status of that and make it a priority for putting it on either one of our leadership programs GitHub pages or probably not protocols.io. I don't think that works very well for like code of this nature, but we'll find a way to distribute it and certainly have a link on the website as well for that. So yes, no problem.
Thank you. And I think that at the OBON meeting, so Becca is at PML and she'll be attending that meeting as well, I think. So you can talk about it in person, hopefully.
Excellent.
Okay. Are there any other questions?
No, I don't see any other.
Nick has his hand up.
Always. Sorry.
Yeah.
I do find this a very interesting topic. So I always have questions, and it's always really nice to see what other people are doing. So thank you so much, Nastassia, for your overview. And I must say, really inspirational as well, what you guys are doing on the West Coast. I wish you were on the East Coast, because that would make it much easier for me. But a couple of things. The first thing, you were talking about mining for different reference material. And I was just wondering if you'd engaged with or checked any of the big sort of global initiatives on reference genomes. There's Darwin Tree of Life and all sorts of things going on that are doing reference genomes, so full genomes. So you might not need to do that. So it could also be relevant for your gap analysis to check what some of these big international projects are doing, to see if they cover any of your gaps. And very often they will have forms or means for reaching out and requesting particular species or groups and so on. I know even the French one does. I know that doesn't necessarily sound very close, but there is French Polynesia, which is covered there as well, which is in the Pacific, even though granted very far away from California. But it's worth checking. But yeah, I would also say, based on my presentation last week, I will get in touch with you guys, because all the reference material is something we're working on at the moment to try and create standards for linking all the references. So if you guys have extensive material already, then we'll definitely be in touch with you to see what you guys are doing, what standards you're using, and how and whether it's available. My question to you is, I mean, you guys have done all this amazing stuff on the West Coast. Have you already approached anyone in government or at the state level about the potential application or use of the data, or the sort of joint products around prediction and so on that you could create on the West Coast? And I'm asking this because in Europe, this is starting to become an interesting avenue for funding, because now we have restoration laws and people are panicking, the countries are panicking, going, well, we don't have half the information, so where do we get it from? So these are interesting avenues for pursuing funding and saying, well, we can fill some of those gaps, or we can help with your management plans. Is this something you've pursued? I appreciate across states this is complicated, but perhaps a place you guys would be able to get federal funding.
Yeah, thanks for that, Nick. Let me see if I can yeah, I'll address this last point first. I think that's really interesting. We're constantly looking for ways in which our like the resources that we assemble, curate, organize could be used for federal or state biodiversity monitoring efforts. I think one of maybe the more likely sources that we've identified as this big source of state funding coming to California that is meant to be specifically for intertidal organism DNA reference barcoding. And so I think we, as part of the reason for developing this guide and focusing around this topic so much in the last year, is to really prepare our members and our cohorts of members to come forward for that source of funding and put into practice all these resources that we've developed around this whole idea. So that's probably the best example of that. And I think federally, I'm not aware of any big federal efforts similar to the ones you're talking about in Europe, where there are, laws around, biodiversity monitoring for restoration, for example. I will say on the West Coast, there's a big push to develop wind energy, offshore wind energy. And there is a lot of activity and talk around biodiversity monitoring before, during, after construction of these offshore wind energy projects. And so that's another big area that we're really keeping a close eye on, and hoping that our sort of existing observing programs could be used to just slide into those wind areas and generate the data that they say they're looking for when it comes to marine ecosystem diversity. So those are a couple of examples that we have focused on, and partnerships that we're trying to build. Oh, one more I'll say is with the Smithsonian Institution, just because they do a lot of things like reference specimen mitogenome sequencing, similar to maybe what you were talking about with the Darwin Tree of Life initiative, where they I don't know the process for that, but they're doing, whole genome sequencing of some select group of organisms, user dependent maybe. So the Smithsonian does similar efforts, and we have not formally sent any samples to them for sequencing, but we talk to them all the time about it, and it's one potential avenue to generate reference mitogenomes for our list of species. So those are, some of the different ways that we're trying to leverage our connections and sort of existing infrastructure in place that could help push forward the things that we talk about a lot in theory and develop guides for. And then you thought you had a second point that I skipped over, something about my reference material, and I'm not remembering that, but I'm happy to go back to that if you remind me.
It was just to let you know we'll be coming your way to find out what you guys have got and standards you're using and so on, to see if we can align and get some inspiration as well. So, and in best case scenario, catch what you're doing. I just wanted to mention on the offshore wind, this has been a good, not necessarily source of revenue, but certainly source of opportunity for us. But we've been doing it with arms panels. And so, well, we do meta barcodes on the arms panels as well as the taxonomy, but we have been doing this as a part of environmental impact assessments exactly for this. So I think there's some publications as well from the arms OBON group, and certainly from our own observatory, where this has been used from companies and so on. So yeah, I would just say that it could be really interesting in those cases to add arms to your list of protocols, because it's a really good one for these, at least from a European perspective, in terms of monitoring and environmental impact assessment. And thank you very much for your extensive answer.
Yeah, thanks, Nick. Just one quick point on the arms. I love those experiments. I think they're so fantastic. I do wonder, I think our offshore wind is going to be in water that's quite deep. And I'm not sure that it would be feasible to put out like panels that can be monitored and collected the way it is for the shallow wind platforms in Europe. But I love it.
Presumably they're going to be floating then.
The idea is that, yes, they will be floating, and then there's some sort of like tether to the bottom, but yes, the platforms themselves will be floating. And so right now I think the focus is really on the pelagic biodiversity, but maybe the panels could actually go on the platforms themselves. I think that would be a cool.
Yeah, I think somebody tried to do that. We're going to try and deploy arms in about 1,000 meters of water in the next year or so. So we'll let you know how that goes as part of arms OBON. So, but yeah, I think you guys are way deeper than that off California. So that's not necessarily super helpful for you guys.
It depends. It really depends on where they're going to end up being. But yeah, I think it would be a challenge to get down to the bottom. But I like this idea of putting them on the maybe on the underside of the platforms or something, and that would be pretty straightforward to regularly monitor. So yeah, great idea.
There was a contribution in the chat going back to, I think, the beginning of Nick's question or comment about the international projects and databases from Kate Sheridan, who's involved with the PECO project that was presented before, saying that they found that the international projects haven't been that useful for their kind of local species, and that NCBI and BOLD have been the most comprehensive databases for North America.
Cool. Yeah, that's been our experience as well. But I will say I don't believe we've formally checked in with the Darwin Tree of Life Initiative, and so they might have some data that are not yet published, but like in the pipeline that we would want to know about. So,
and the Darwin Tree of Life is a UK project, isn't it? Is it?
Yes, it is UK funded. Yeah. But I mean, somebody said they already checked with BOLD. What's great with BOLD is that they normally publish the metadata even before they have all the actual species. So at least you know where samples have been taken from. So it should also give you an idea, at least on BOLD, whether there's anything coming from your neck of the woods.
I had a question, Nastassia, about data. I may have missed this in your presentation, but are you talking about kind of standardizing data and metadata between the different projects that are involved, and where ultimately where the data is or will be submitted?
Yeah, so I think right now we're kind of in a phase of developing resources and encouraging the adoption of certain, standardized methods. We have such a range of programs and members that we cannot, mandate for anyone to follow some protocol when they've been doing a survey for 5 or 10 years a certain way already. This is not up to us necessarily, but what we can do is provide these resources, demonstrate why it's valuable to use them, and then I think the hope is that over time, as these meetings proliferate and our conversations get deeper and more frequent, that ultimately people will gravitate towards using tried and true best practice, verified protocols. And so that's sort of the long-term vision, I would say. And then similarly with data publication, we push very strongly for not only depositing raw sequence data, but also now taxonomy tables and metadata onto OBIS. And so this is something every time we, give a talk or provide guidance, we talk a lot about the importance of doing that for biomonitoring programs. And so, we hope to sort of lead by example on this front and make it just a standard thing, just how uploading raw sequence data to NCBI is like a standard thing that everyone does. We hope to make it the same way for depositing actual diversity information onto OBIS and GBIF. And so I say that's the phase we're in right now. Without any real authority to mandate anything, we can lead by example, we can demonstrate the effectiveness, and then hopefully, generate the cohesion that way.
Yeah, sounds great. There's been a lot of discussion and information coming out of these project presentations about resources, that are being shared and that are being made available. And I don't know if Margaret wants to comment on the OBON capacity development working group and plans that they have, but it always feels like there's a lot of things that we could be tying together. And so OBON, we're trying to make resources available on the OBON website, and I've made a note to check what you have on your website and make sure that we're kind of cross-referencing as well. But ultimately, we want to share these resources and perhaps use some of them as training materials for people who are coming into eDNA, who are new to the field, getting started with setting up an observatory, or colleagues in developing countries who want to get started. So we'll definitely we'd definitely like to talk to you about sharing the resources that you have, and the ones that you mentioned in your presentation about reference barcoding guides and so on. Margaret, do you want to add anything on that?
Yeah, the capacity development working group is working on some in-person workshops, but they are only available to such a restricted number of people that we decided that in concert with that, we were going to make an effort to collect the online training, that videos that are available, and to sort of curate those. So, for example, there might be an online video regarding sample methodology for a particular style of analysis in a particular region, but that is really great for a beginner. And something else that is a discussion that might be something for, you know, somebody who has a lot of experience. So that as we collect things on specific topics, that then we would write just a brief guide to those. You know, here are 7 videos on sample kits, and these videos are for, you know, kits that might be applicable for citizen scientists. These are kits that are applicable for experts who are, you know, trying to do very, very precise work that's going to be published, and then try to build up that resource so that at every level you would be able to say, oh, I could get started by looking at these 7 videos and you know, approach the problem that way. And I also noticed, Nastassia, that you were talking about your manual. And so we're also thinking about that as well, that a lot of you have manuals that you've developed for a particular region, for a particular type of work, and that it would be helpful for us to have a common resource on the web for manuals. So we're Sophie and Liz are in the process of reaching out to all the projects to say, you know, it doesn't even have to be something that you generated, but oh, here's a great video that was done by Ocean Teacher, and it's available on such and such topic. So as we, you know, please help by responding to those requests.
Thanks, Margaret. I don't see any more questions or hands. So I think maybe we will wrap this up. And please stay tuned for information on the next 2 webinars, which we're due to have in the next couple of weeks. So we have a few more kind of observatory type programs that haven't yet presented, and we have a few programs that are working kind of with fisheries and policy and citizen science. So we've kind of grouped all of those together to have a more end user focused session. But we're just finalizing dates and times for those. So we'll send that information out very soon. So yeah, please register for the next couple of webinars, and thank you very much for joining this one, and thanks once again to the speakers for their great presentations, and thanks for all the lively discussion as well. It was great to see the enthusiasm.